Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Kia ora koutou and welcome to another episode of Sigest. My name's Rebecca Bloomer and we are joining you from our Lincoln site today, where I have the great pleasure of introducing you to Andrew Twidle, who's a scientist in our ecological pest management group. Welcome, Andrew.
Yeah, thanks, Rebecca.
And today we're going to be talking about a project that was recently funded in the group that Andrew is leading, funded by MB through the Endeavour Smart Ideas program.
That's correct.
Where the group are looking at developing pheromone-based tools to control redback spiders, which I only very recently discovered were even present in New Zealand, let alone not that far from my office. Andrew, can you tell me first, how long has there even been a redback spider in New Zealand?
Yeah, well, I was like you, Rebecca. I was actually surprised when I found out about them initially. They came into New Zealand in the early 1980s, and they've established a couple of populations in New Zealand, the main one being in Central Otago in the Cromwell region.
And the other one?
And there's another population in the North Island in around New Plymouth.
Sure, a little further from me, which I'm comfortable with, if I'm honest. And so since establishing here, they haven't spread too much further, obviously.
No, so we've been pretty lucky, really. So they established in a pretty unpopulated area. So redback spiders, they love to live around human habitats. They do really well associated near humans because we provide buildings and things that are nicely sheltered and warm because they like those sort of conditions. And obviously Cromwell's not that highly populated, so they probably came in on the bottom of a pallet. On the pallet was probably a female that was pregnant. She laid her eggs, and then the populations slowly established. From there, just to give you a comparison, so they are around Cromwell, Alexandra, Wanaka, that sort of. We don't know exactly how far they've spread, and that's another thing that this potential tool will allow us to do, is to see how far the spiders have actually gone. But just to compare that, so they invaded Japan as well in the 1990s, so about 15 years after New Zealand, and they came into Japan in Osaka. Now that's a really highly populated port city, and since then they've spread to 45 out of all of the 47 districts in Japan. So they've just gone poof through the country. So we're really lucky that they haven't done the same thing here yet, but there is that potential.
Sure. And whilst acknowledging we're fortunate that so far that hasn't happened, what are the problems that they've brought along with them so far?
So the main concern is they're obviously a threat to human health. So
and specifically, having met some of your spiders recently, how much of a threat?
Quite a threat, really. So they're one of the most venomous spiders in the world. Most spiders have venom, and they use that to kill their prey. Well, pretty much all of them, except a few species, have venom that just kills other insects and spiders. So it's not a threat to us bigger animals. But the redback, it's part of the infamous widow genus of spiders, and they produce a special kind of toxin called alpha-lactotoxin, which actually targets larger animals, so animals with a backbone, and that includes us, because they can catch in their webs because they've got really strong silk. So their silk is one of the strongest out of all the spiders in the world. It's stronger than steel by weight, and they can catch things like mice, lizards, and even snakes in their web, in their native habitat. So they have venom that can kill those animals with a backbone, and unfortunately for us, that includes us. And
I'm sure that poses some interesting challenges in the lab.
yes, it can involve a bit of health and safety challenge.
I'm envisaging some paperwork, among other things.
Yeah, there is a wee bit, but we take it really seriously because the studies that have been done on the venom, it's actually more toxic than rattlesnake venom. But thankfully for us, when they do bite, they generally, a lot of them don't actually inject venom because it's just a defensive action rather than a prey, trying to kill prey, so they're defending themselves. but they can inject venom, and it is obviously dangerous. And New Zealand hospitals now do stock Redback anti-venom, which is something I didn't know until I started this project.
The more you know.
Yeah.
Tell me a little bit more. So as well as being a threat to human health, they're posing a few pretty key challenges to some of our native species too.
Yes, so they pose a big threat to a lot of our native fauna, and the most threatened of those at the moment is the critically endangered Cromwell chafer beetle. So this is the only beetle in the world with its own nature reserve. So down in Cromwell there's a nature reserve for the Cromwell chafer. It's the small brown flightless beetle, and it only lives in these certain sand dune area of Cromwell. There's about 3,000 of these beetles left in the entire world, and sometime in around the 2010s, we don't know exactly when, they invaded the chafer reserve, and a student from Otago University who was doing a project around there, she found redback spider webs in the reserve with as many as 10 individual chafers in one web. So over those couple of years she was recording in there, she found hundreds of dead chafers at the hands of the redback spider. And now dock rangers manually patrol the reserve, manually removing webs and squashing spiders. So that's a pretty arduous task because they're pretty well hidden and cryptic. So that's a bit of a challenge. So that's one of the areas where, as I said, we're trying to develop a tool that will attract these spiders so they're not so hard to find and catch.
So on that, could you tell us a little bit more about your SMART idea?
So just to give you a little bit of a background on some of the work I do, so I'm a chemist here at Plant and Food, and the main work I do is on the identification and synthesis of semiochemicals. Now semiochemicals sounds like a big nasty word, but it's really just looking at signal chemicals that plants and insects use to communicate with each other, and then we try to develop environmentally friendly pest management tools based on those communication compounds, because it allows us to send out a targeted message to what the particular pest rather than a broad spectrum thing that just kills everything. And that's what we're looking to do with the spiders, with this smart idea.
On that, so semiochemicals, you mentioned they're really targeted. They would tend to be very species specific.
So there's a type of semiochemical called the pheromone, and that's compounds that act between members of the same species and gives direct messages that they use to communicate directly with each other. And one of our collaborators at Lincoln University, Cor Vink, who's the New Zealand spider expert, he's the one that sort of initiated this project, and he came to me saying, we can see that the redback's using some sort of airborne signal. Can you help us, as me being a chemist, identify what that is? And that's how this project got started.
Sure, and something that's quite unique about this project, as I understand it, is that identifying and using pheromones as lures is a reasonably common approach in insects, but this is a first for spiders, right?
Yeah, that's right, Rebecca. So the first insect pheromone was found in 1959, that of the silk moth, and since then people have realised how powerful pheromones are, and they've been using them in horticulture to try and protect crops from insect pests. And since that 1959 discovery, there have literally been thousands of insect pheromones identified worldwide, and many of these have been used in pest management. Now, spiders, on the other hand, they haven't received much study, and to my knowledge, there's only been 15 spider pheromones identified worldwide through human history, and currently none of them are used pest management tools. So we're trying to take forward, our ability and knowledge from using these in insects and apply these to spiders and identify the signal that the redback spider is using, and then use it to specifically trap the redback spider. And where we're in a really unique position with this project is that with insects, a technique that's used with pheromones is lure and kill. So that's where we put out the sex pheromone that the female produces to attract males, or we'll put out a copy of that pheromone that the female makes and lure the males to a trap and kill them. Now that works pretty well, but one of the downfalls of that technique is that, say we have 100 males in the environment and our pheromone trap catches 99 of them, that one lucky male that's left, he can go around and mate with all the females, so you don't get a massive population drop. But with the redback spider, they have a really unusual mating behaviour. So when the male and female mate, the male sacrifices himself to the female and she eats him. So
Brutal.
a male can only mate once. So if we catch 99 of the males to our trap and there's that one left in the wild, he'll only be able to mate with one female. He can't go around and visit all the females. He can only mate with one. So I think this is a really good opportunity for a powerful technique to be applied to the spider control.
Sounds good. So I think we've probably covered some of it, but I slightly derailed you starting to outline the project by asking about semiochemicals. So tell us a little bit about, I guess, the broad aims and approaches of the project. Where do you even start with a project like that?
Well, our broad aim for the project is to develop a tool that could be used to eradicate redback spiders from a particular area. Now, we've got a fantastic team on hand. So we've got Corv Inc. from Lincoln University, who's our spider expert. We've also got a student under Core, so Nicola Sullivan, who's doing her PhD on spiders. She's helping us out as well. We've got Meikura Arahanga. She's our cultural lead and community outreach specialist. We've got Tara Murray from DOC, who's our chafer expert, and other general threatened invertebrates experts from the area. We've got Tom Sullivan, who's like a trapping guru. He can design anything with his 3D printer, and that'll be a big part of the project as well, designing a trap that's selective as well for these spiders. And then there's me, the chemist, who'll be actually identifying and making the pheromone.
So what's the first step?
Our first step was last year in the early summer. The whole team went down for a bit of team building in Central Otago and collecting spiders, because we needed to establish a lab colony of the spiders. So what we're aiming to do is, we know from the original work that Core did at Lincoln University that the female silk is attractive from a long range to the male spiders. So generally speaking, these are really small animals and they don't produce a lot of these pheromones. And unfortunately, most of our chemical instruments need quite a bit of material to be able to identify what these compounds are. So we need a lot of the material. So we went down to Central Otago, collected a lot of spider, and over the last 3 months, 4 months, we've been harvesting heaps of silk off virgin female spiders, which we know is attractive to the males. I'm using that silk to try and identify the airborne signal that's coming off it.
So harvesting heaps of silk implies that establishing that colony has gone really well,
Yeah,
actually.
so
Have there been any kind of challenges with getting them happy in the lab?
No, so they're really happy in our lab. We've got about 800 of them in our lab right
I
now.
have really mixed feelings knowing that there are 800 venomous spiders not that
They're
far
all
from my office.
individually housed and double contained, because obviously we don't want any to get out, because there aren't any that we know of yet established in the Canterbury region. So we're really careful about that.
Yeah. But that's gone well by the sounds.
Yeah, they're thriving in the lab. The only problem we're having is that with the males, for some reason, seem a bit harder to rear than the females. So we're having a bit of challenge getting as many males as we'd like, because we're trying to build up the males as well, because we'll eventually test them. So at the moment we're identifying the chemicals coming off that female silk, but then we also want to test those chemicals in like little Y-tube or arenas to see which ones the males like the smell of the best. Right. So when we're developing our lure, we can get the optimal attractive blend for those males that they find irresistible.
So colonies happily established, loads of female virgin spiders to collect silk from.
Yeah.
Males so far a little more challenging, but
yeah,
coming
not
along.
so many, not as many males as we'd like, but our numbers are improving. It's just a lot more manual handling and feeding than we were hoping for. So
manually feeding venomous spiders is a real interesting proposition.
Yeah, so we have, as I said, each individual spider has to be fed weekly, so that's a lot of...
This is quite a big job, I imagine.
It is quite a big job.
And And silk collection's going really well. Have you been able to collect enough yet to start doing any chemistry?
Yeah, so I've already started identifying compounds. So we've got it's really exciting in that phase actually. The virgin silk looks completely different odour profile wise compared to the mated females and the sub-adult females. So there's 3 extra compounds that the virgins produce that the others don't. So
That
we're in
narrows
the
it down.
process of identifying those compounds at the moment, and then we'll start testing synthetic versions of those with males to get the right ratio for their attraction.
Sure. So I'm no chemist. Can you tell me in sort of simple terms what it is that you do to detect and to kind of understand what the compounds are?
So typically in chemistry, if we're wanting to identify something, there's this marvellous machine called an NMR, and you take a quantity of that compound, put it into that machine, it'll pretty much give you the spatial arrangement of atoms in that compound, and you pretty much have a good idea of what the structure is. Unfortunately, because we're dealing with such small amounts of compound from these really tiny creatures, we can't use NMR, so we're having to use mass spectrometry, which doesn't give us as much information, so there's a bit more good old-fashioned detective work in identifying the compounds. So we get a molecular weight, so we know sort of how big the compound is, and we sort of get an idea of what the atoms are, but we don't get that same idea of connectivity. We sort of have to work that out for ourselves, so it's a bit more challenging in that aspect. But as a chemist, I find that fun, and it's a good puzzle to try and solve. And luckily for us with the spiders, the molecules look pretty small, so they aren't that complex. Generally, the bigger the molecules, they obviously harder to identify because there's more options that they can be.
So you think you've got 3 compounds at the moment that are your candidates.
Yep.
Presumably you're making a bit of headway figuring out what they actually are.
Yes.
And at some point, the next step is going to be synthesizing those
Yep.
synthetically. Again, this is one of those things that as a non-chemist kind of blows my mind. So can you tell me at a kind of high level, how does that work?
The best way I like to explain it is, well, it's sort of like baking a cake really. You're just taking a number of ingredients together, combining them to make a finished product, and that's sort of what we do as a synthetic chemist. We're taking smaller ingredients and putting them together to make a bigger compound. Almost like building blocks with Lego really, so just adding little bits together to make a bigger bit, if that sort of makes sense.
Sure. So you don't quite know what you're building yet, but you're not foreseeing that being a
We've got a
hugely
pretty good
tricky step.
idea, and to be honest, it looks pretty straightforward, so I don't want to get too confident. I've had that blow up in my face before, but things are looking good at this early stage. I'm confident we can crack the structures.
Cool. So in terms of testing among your 3 compounds, which one might be, or which combination
I think
might
it'll be a
important?
combination of all 3. It'll just be getting those, because often with pheromones, closely related species will use some of the same compounds, but then they'll have different ratios of those compounds as their signature for their species. So what I'm expecting is there'll be, those 3 compounds will be in a very sort of defined ratio for the redback spider. So we'll need to get that airborne signal just right to be highly attractive to the males.
Sure. And how do you go about testing that? What does it look like?
So our initial steps there will have, like, if you can imagine a large Y made out of glass and it's hollow inside, so it's effectively 2 tubes coming together into 1 tube. And in each tube there will be a different odour. So there will be, on the branch of the Y on the left there will be one odour, and on the branch on the right side of the Y there will be another odour, and then in the middle of the Y there will be the spider, and then it'll get both of those compounds it'll be blowing over it, and it'll make a choice of which side smells the most attractive, and that's how we'll sort of narrow it down initially, try and get that blend get the spiders as attracted as we can to the blend, and then we'll go out into the field. So one of the things I'm doing on the side at the moment is actually contacting vineyards in the Central Otago area, because we know they are a problem in vineyards. We've spoken to a few vineyards already, and some of the vineyards have large numbers of these spiders, and they actually spray for them. I didn't even know that. So they're really excited about the work we're doing as well, and they'll be great to have on board for field sites to trial these compounds.
So I would really like to come back to that point actually in a minute, but I'm still kind of fascinated about this Y testing business. Do we know anything about how the male spiders are perceiving these pheromones?
So that's a really good question, Rebecca, and I'm sure Core does, who's our spider expert, but me as the chemist, I'm not exactly sure. But I have seen them, I have done a couple of trials just with female silk in the Y tube, and they kind of wave their front legs around when they're in the air. So I'm not sure whether they're like using a sensor on their leg to detect that pheromone, or whether they're trying to wafting the pheromone into their face with the leg. I don't know, but they certainly do something with their legs when they smell something of interest.
Yeah, and are you seeing with those initial tests, like it's presumably pretty clear that
yes.
the webbing is attractive?
Oh yeah, so we've done Y-tube trials, and only really preliminary stage at the moment, but we've had virgin silk in one arm and the silk from mated females in the other arm of the Y-tube, and it's gone straight to the virgin female silk, not the mated female silk. And again, from our chemical analyses that we've done of the silk, we can see why, because there's three really distinct large amounts of these three compounds in the virgin silk that seem to be absent from the mated silk. So the female just clearly stops producing these compounds once she's mated. So there looks to be a clear signal there. It's just us getting it right is the key to this project.
So coming back to you talking about doing some work in vineyards, and a point you made at the beginning as well. So you're working with a few different collaborators, and at least one of those has a real focus around community engagement. Can you talk about that a little bit?
Yeah, so Mai Kura Arahanga, she's our community outreach expert, and she's been great help to our project, liaising with local iwi and just getting local community on board for the project. So again, it's early days yet, but everyone that we've spoken to is just, really supportive and, excited really about the project. So and thinks
fantastic.
it's a good idea that we get in and do something about this before it becomes a bigger problem, because it's only a matter of time before they do spread. And by us developing a tool, we give New Zealand a good chance to respond if this comes into one of our main centres to be able to wipe it out.
That's another point, isn't it? Is that at the moment they're localised in two places, but if you had an incursion somewhere a bit more populated, things
could be a real problem. And the other thing is, at the moment they're really cryptic little creatures. Like, they're actually quite hard to find in the wild. Like, we know there's a lot of them out there in certain places, but they're still just well hidden. They hide under rocks and bushes and things, and they're hard to see. And one of the things that this pheromone tool will allow us to do is we can put traps out in certain locations all around the country and just see actually whereabouts they are and where they've spread to, because at the moment we've got no real way of easily finding them.
Yeah, so you've got a monitoring tool as well as a pest management one. Yeah, that's the great thing about this.
So as well as having the pest management tool, is also that monitoring tool, and we can get a much better idea of where they've established and where, if they're getting close to one of our main centres and things. So
Yeah, that makes a ton of sense. You mentioned a little bit ago that part of the project is around designing just the right kind of trap. What's that about?
Yeah, so while these pheromone signals are species specific, when we're making a trap that can kill something and we're putting it out in an area where there's endangered insects, we don't want any accidental chafer beetles stumbling in there and just getting killed by chance when they're so precious. And the other thing is, while it's not a problem in Central Otago, it will be a problem in the North Island in New Plymouth where they've established, and that there's likely to be katipo populations as well. So katipo are also a widow spider. They're New Zealand's only venomous native creature that I'm aware of. But they're endangered as well, so we don't want something that they could find a wee bit attractive in coming into that. And so we want to make something that's exclusive for redback spiders. And we are, again, lucky in this project in that there's a real difference between the male and the female spiders, and the males that will be coming to our traps are really small. So if you envisage a female redback spider, she's a big spider. So her abdomen's nearly the size of my thumbnail, and then she's got all her big legs as well. She's quite a big spider. The male, in comparison, his abdomen's about the size of a pinhead.
Right.
If you saw the two beside each other, you wouldn't even think they were related. That's such a size difference.
Yeah, so that gives some
And
opportunities,
appearance difference as
presumably,
well.
to base a size sort of exclusion.
Yes, a size exclusion trap. And also, again, we need to spend some time in the lab, and this is where Tom will be a master, like looking at behaviour as well of the spider, seeing how they approach the trap and how they look to go into the trap. All that sort of thing will help in the design of the trap, because we want something that doesn't put them off going in. So they'll be wary of a trap, as in just it's not their usual. We've got to try and make something that appears natural,
not
unnatural.
Kind of enticing enough.
Yeah.
So that's an interesting point you've talked about, and actually I've had the pleasure, in inverted commas, of meeting some of the spiders in your lab. The girls are obviously making webs.
Yes.
What are the boys doing?
So,
where are they hanging out in the wild?
So in the wild, when they're first born, all of them generally stay in their mother's web to start with, because they are too small to be prey for the mother. So she doesn't, and they just scavenge off things in her web. As they grow, the females obviously grow a lot bigger than the boys. So once the females reach a certain size, they go out on their own and establish their own webs. Again, this is probably a better question for Core, the spider expert, but the males, I think, stay in the web a bit longer than the females, and then they can actually make wee webs of their own, but obviously they'll catch a lot smaller prey than the big girls.
Yeah, it's interesting. I hadn't sort of twigged to the fact that there were probably some quite gendered differences to their whole lifestyle, actually. You mentioned wanting to design something that will hopefully exclude katapo spiders. But we've also, I think, touched on the fact that quite often it's specific ratios of chemicals and things that are important. So do you think that's realistic to be so species specific?
When you've got closely related species, there will be a wee bit of cross-attraction with chemicals, just because they'll probably have some of the same chemicals in their pheromone blend. And if the boy's desperate for a female, he'll go, well, maybe.
Good enough.
This might be close enough. But one of the things, again, where we're lucky with this project is katipo don't have such a difference in size between their males and females.
Right.
So the male katipo is a lot bigger than the redback male.
Okay.
So we think
Suggest there might be some mechanical differences at play there too.
Exclude just based on size alone there.
Do we know anything about whether the two species hybridize? Is that a potential threat as well?
That is a potential threat, and I think there has been some preliminary work by Lincoln Uni, but I'm not really fully aware of exactly what's been done and what hasn't been done. But I know there is reports of them being able to mate. So,
Andrew, at this point you have got a happily established colony of spiders. You've got a pretty good idea of the chemicals that you think you're going to be synthesizing, and that's hopefully going to be reasonably straightforward. As you said before, touch wood.
Yeah, touch wood.
What are the next steps for the project beyond that?
Yeah, so as I mentioned earlier, once we've got those synthetic compounds, we'll do the lab Y-tube trials to try and identify that blend. But then once we've done that, it'll be taking the lures out into the actual field. So that'll be vineyards initially, and then hopefully the Cromwell chafer reserve after that. But it'll be just getting that signal right in the field, so it's as attractive or more attractive than the virgin females.
Sure. Do you foresee any particular challenges with that? You've already obviously got some vineyards on board to provide testing space.
Yes, we do. But for me, as a chemist, the big challenge will be getting the right mixture in the air for the spiders to come to. So when we've got those, as I mentioned, those 3 virgin compounds, if we load those into a dispenser, we need them to come out of that dispenser at the right ratio, otherwise it won't be attractive to the females, and that'll be a big part of the project, getting that dispensing system that gives us the same signal that a female's giving. So that'll be a challenging part of the project, and I can foresee that being difficult.
Yeah. Yeah, lots of things to potentially test within that, though, I guess. How are they dispersed? They kind of just windborne? Or?
Yeah, so they're really small molecules. They volatilize and just as a gas and travel through the air as an odour plume, and the spiders come towards that.
Is that kind of stuff quite temperature sensitive too? Like, would you imagine a hot Cromwell day being different than a frosty morning?
Yes, and that's a big part of it. We will only be realistically trapping over the summer months, I would think, maybe late spring and then summer, because they won't be active. It'll be too cold for them.
Yeah, sure. So there's some stuff around spider kind of lifestyle choices there too.
Yes, yeah.
Makes sense. Assuming that this all goes swimmingly, and I'm sure it will, how wide do you see the kind of future applications of this being?
Well, I think there's a lot of opportunities for this. I mean, as I just mentioned earlier, Japan's been hard hit by the redback spider, and I'm sure they'd absolutely love a tool that would be useful in controlling that spider. And even in Australia, the redback started off before Europeans got there in a small little area of South Australia, but with increasing in human habitation, they've spread all around the country and are associated with a lot of people's houses and around people's places, and I'm sure there'd be a market there as well.
Yeah, sure. So they're native, but they're still actually quite problematic.
They're a real pest, as well. And I mean, this is just scratching the surface. This is just redbacks. I mean, that whole widow genus of spiders, they're a real health threat worldwide. I mean, there's a widow spider in each continent, pretty much, other than Antarctica. So they also represent biosecurity threats. The world could be our oyster, so to speak, and that, no one's really looked at controlling spiders with pheromones before. And as I said, I think it puts us right at the forefront of spider chemical research and gives us a really good opportunity to be first through the door, so to speak.
Yeah, that's super cool. What an exciting space to be in.
Yeah, we're stoked that we got the support for the project, and as I said, just the opportunities.
That's awesome. Well, I'm so pleased that at least initially things seem to be going really well. Hopefully that remains the case, and maybe, a little bit further into the project we can catch up again and hear how it's all going with deploying in the field as well.
Yeah, that'd be great, Rebecca.
Cool. Thank you so much for your time today, Andrew, and thank you as well to our listeners for joining us. Hope you've enjoyed this as much as I have, and hopefully you're not any more worried than I am about the potential for redbacks in your office now. As ever, thank you very much for listening to Sigest. We do really appreciate if you can subscribe and rate our podcast. It helps us increase our, I was going to say readership, that's not the right word for a podcast, our audience. So we do appreciate if you can take the time to do that. Until next time though, mā te wā.